Well, thank you for the kind introduction and invitation, Jane. So I'm Nadir Kaplan. I'm an assistant professor of physics at Virginia Tech, and so I have actually a broad interest of a broad research agenda. But today I'm going to focus on the biophysical aspects of our recent work, and specifically mechanobiology of mesenchymal cell migration, and how we can actually take different modeling approaches, ranging from like holistic whole cell modeling to more conventional physicists like reductionist approaches. I'm a physicist by training. So with that, let me first. I should be able to. Okay. Yeah. So let me actually first give a brief overview of my research before I dive into the topic of mesenchymal cell migration and the mechanobiology of it. So I'm the head of the biomaterials theory group at Virginia Tech Physics. So some of the recent research projects that we have been tackling are on this slide. So on top left, what you see is here, I'm just switching. Okay, so the laser pointer. So here, what you see is a membrane of a lipid bilayer on a surface, on an aluminum surface, which is initially totally wetting in the presence of calcium crosslinkers. But as soon as chelation occurs, like calcium is dissolved from the interface between the aluminum and liquid bilayers, these types of bubbles are observed to grow. And so we actually took this question in the context of origins of life with my longtime collaborator, Irep Gözen from the University of Oslo, Norway. And so we asked the question if this system can actually be a model for compartmentalization in model protocells. And here, like our contribution as physicists were essentially, can we understand the growth of these bubbles, which are different compartments and different protocells in the model system, as a growth through an elastrohydrodynamic instability? And then what should be the onset of that instability? So that's the approach that we took. And of course, there's a lot of questions there still. Like what we did was a deterministic modeling, but there's a lot of stochasticity going on associated with thermal fluctuations. So that's a brief overview of that. So we also focus on gel mechanics, but specifically the mechanics of chemically responsive hydrogels. And the main question that we are asking here is, as in biological systems, can we actually incorporate reverse feedback from mechanics to chemistry? So when we say stimuli responsive materials, we really mean that there are like some stimuli that excite materials, and then the materials essentially, give a response. But then the reverse feedback would be, as soon as a material gives a response, can it also modify the initial stimulus? So that's the context that we are taking. That's the overarching approach that we are taking. But within the context of hydrogels and elastomers, so the questions here would be like more specifically here, we are considering if gel deformations can be made super diffusive because they are known to be diffusive. So that's a fundamental limitation in gel swelling and contraction. So we are having a recent publication about that we published last year, and then we are also having some ongoing work. And we also recently started doing experiments. So I'm a theorist myself. I worked in experimental groups before as a postdoc and a graduate student, and I still did theory back then. But now, like finally, I'm also designing and running my own experiments. Another question that we recently tackled was the deformation of viscoelastic elastomers and how we can control their deformations and rates to be able to come up with complex configurations, but also not just that, like can we actually come up with multiple target shapes? So that's a question that we are always having in mind. And so I'm a theorist, I'm a physicist. So that actually, so my background is in statistical mechanics and then in fluid mechanics and in solid mechanics. So that actually all those background of mine eventually drove me to the realm of neural networks, um, which is of course now becoming a very fundamental tool, just like conventional simulations that we have been running. But we have been asking the main question that, given that neural networks are known to be universal function approximators, and given that they actually allow us to take derivatives to, to an accuracy that we never could achieve before on computers with numerical derivatives, can we actually um, 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 design neural networks as non-convex optimizers to find the global phase diagrams or stability diagrams of dynamical systems or many-body systems? Like, for instance, how can we characterize the fixed points or or other types of singularities like bifurcation points in dynamical systems, etc. And then a recent question. So I also part of my background is in differential geometry and especially in morphogenesis. So dating back to my graduate studies and my postdoc. So a recent question that we have been asking is the dynamics and the dynamics and mechanics of curves, of elastic curves confined to non-Euclidean surfaces. So when I say non-Euclidean surfaces, what I mean is really, all kinds of curved surfaces that are simply connected, meaning the topology is simple, but we don't have any limitations on geometry here. We don't prescribe shapes. We can essentially tackle any shape. And then we can also allow the shape to be determined by the dynamics or the energy of the system. And here the main question is that if we can we develop a general differential geometric framework for elastic curve dynamics on curved surfaces? Like, for instance, it could be all kinds of filaments, including polymers that are absorbed by surfaces. It could be at the cellular scale. It could be at a typical fluid interface, like the legs of a water strider, and then how it bends the surface, and then what will be the configuration of the filament on the surface and the thermal fluctuations of that. So those are the main questions that we are addressing right now in my group. But with that, I actually want to shift to the focus of this talk, which is mesenchymal migration and specifically the mechanobiology of it. So here, what you see is fibroblasts, and the green labels, fluorescent labels, are denoting the actin filaments here. And so, what caught my attention when I first started looking at mesenchymal cells was how should I say, how dirty the system is. Like, for a physicist, we look at continuum mechanics, we look at like smooth configurations, etc. But here, what we see is like these like very pointy structures, where it's very difficult to apply continuum mechanics. So of course, there are like models, active matter models, active matter nematic and polar models of actin configurations. But here, when you consider the migration of that, one needs to keep in mind that a mesenchymal cell, in fact, migrates through adhesion on surfaces. And what it does is it essentially extends an arm like a gecko does. But these are here like Philopodia or Lemetopodia. So these are Philopodia actually. So those are actually like very like pointy and almost creating a singularity both in the configuration of the membrane and then also the cytosol inside the cell. And so these arms extend, so they adhere and then they pull the cell forward. And while this is happening, the cell also needs to have a simultaneous retraction on its back. And if you consider the motion here, the mechanics of that, and especially on a 2D surface, which is in fact an in vitro setting, not an in vivo setting, you would actually immediately realize that that migration must be associated with shear stress between the surface and the cell. And then, so here, if we just look at a cartoon view of how mesenchymal migration occurs, it's basically front extension and rear retraction. So what happens is, so there's strong adhesion on the front, which is associated with strong actin polymerization. And then there's some strong retrograde flow at the back, and this retrograde flow is associated with depolymerization of the actin on the back. And this retrograde flow is also a result of weak adhesion at the surface, at the back of the cell with the surface. But all this like weak adhesion, strong adhesion, and then how the focal adhesion points form in the first place is in fact governed by chemical signals inside the cell, like raw GTPase molecules or other types of molecules such as CDC42, etc. So it's a very complex setting. It's not just mechanobiology; it's also reaction and diffusion and going on, but not just diffusion. Also, very likely active transport. I mean, active transport is known, but also passive transport through fluid flow inside the cell. So with that, our motivation has become over time, within the context of mechanobiology, how can we understand the influence of extracellular matrix stiffness and viscosity on cellular motion, and especially spreading and migration, which are actually tightly intertwined, as I will elaborate on shortly, but also what would be the effects of constraints or of confinements on single cells, like steric barriers from surrounding tissues or fibers or other cells, etc. So with that, what actually strikes me most about mesenchymal migration is it's a multi-scale process. It's actually very difficult to isolate different processes when you want to ask the question, how does a cell migrate? And in that, actually, for instance, subcellular processes like focal adhesion dynamics, the protrusion growth, and then the actomyosin contractility, all these are coupled. But also cell level chemical dynamics, how the reaction and transport dynamics of intracellular biopolymer network like raw GTPases essentially regulates migration, and then how actually it polarizes cell in the first place, but also cell level mechanics, because there's a structural integrity of the cell. I mean, you could see it as a cable truss structure or a miniature building where everything is actually is working in great harmony. And then at the level of the cell, the extracellular matrix and cell interactions, I mean mechanical interactions, but also cytoplasm viscoelasticity, and also how like the nucleus is essentially tightly bound through stress fibers and microtubular networks to what's going on at the periphery of the cell. So, and as I mentioned before, confinement is also very important because that's going to mean contact mechanics between the cell and the walls of the confinement. So with that, so this actually drove us to a somewhat holistic modeling of the cell. I'm actually a bit cautious when I use that word because it's holistic for a physicist. It's not holistic for a biologist. I mean, if a biologist looks at our models, she would find it like very, very simple. But for us as physicists, what we want to do is we actually want to include still as few things as possible, but not simpler than what we should try to explain here. So that actually forces me to call it a holistic model in the context of physics, simply because we here model the cells as cable truss structures in the context of vertex-based modeling. So that's what we do. And this model here is actually thanks to my previous postdoc, Wenya Xu, who is right now a faculty member in Nanjing University of Astronautics and Aeronautics in China. So he's a civil engineer by training. So he got his PhD from Rice Civil Engineering. And then when I asked him, like, why should we not build a cell? And then as a civil engineer, he literally built a cell, I mean, which is, you know, a computational cell. I mean, I call it actually a Tamagotchi. So we have this, you know, this cell living in our computers, and it actually allows us to verify, quantitatively verify experimental findings, but also at the same time make new predictions, which I will detail shortly. But also on the other hand, so this cell model, when we were trying to build it in the first place, the holistic model, it actually forced us to ask simpler questions simply because we couldn't find anything relevant in the literature. Like, for instance, when a protrusion grows, how does it know when to stop? Or in other words, like more technically, what should be the feedback from the size and shape of the protrusion back to the signaling or to the interior of the cell, so that the cell decides that the protrusion must stop growing, and what should be the mechanics of it. So with that, we actually like isolated that question and then took a reductionist modeling, more physics approach. And in this context, I will actually talk about the fluid mechanics near kinks, near singular points, like the tip of a triangle here. And that's actually a work that is currently being done by my graduate student, Mohammed Hattab. So I'm going to show you some preliminary results on that, and I will also motivate where we are going from here. So by the way, I'm happy to take any questions whenever you like. So please feel free to unmute. So with that, let me first focus on the holistic modeling part of this talk. So the main question when we first thinking about mesenchymal migration and the mechanobiology of that would be actually a relatively how should I say, a more mechanobiological question than many other things that we now can tackle with this model. And in fact, the question was the stiffness-dependent migration, known as durotaxis or anti-durotaxis. So that actually brings a migration paradox for isolated mesenchymal cells. Like, for instance, there are cells that undergo migration from soft regions to stiff regions in a tissue, which is defined as durotaxis. But then these same cells or other types of strains can also take the opposite route. Like, for instance, this happens in metastasis for cancer cells. They undergo anti-durotaxis, you know, from stiff to soft. So, like, for instance, stepping back in the context of durotaxis, there are glioblastoma cells and breast cancer cells that can do that. But these breast cancer cells, they can also switch to migrating against stiffness gradients called anti-durotaxis. But they can actually also decide to be insensitive to the gradients. So when one sees this, the temptation is to associate a higher level intelligence and decision making to the cell, but we can actually now show that we can understand this purely based on mechanical grounds and on passive feedback between the cell and the underlying matrix. So of course, like with that, another question is, I will always, bring the component of confinement in the question, simply because whenever there is stiffness-dependent migration, and then whenever you have confinement in the picture, then that stiffness-dependent migration will either qualitatively or quantitatively be modified. So with that, like the overarching question that I want to at least address partly here would be how do the mechanical interactions between a cell and environment control the migration behavior? So for that, so this slide is so there's some math and schematics here, but I will try to simplify it for you. So the multi-scale whole cell theory that we build is really a polygon-like structure. So this is an octagon, but we actually kept a hexadecagon for convergence purposes. So this is schematic where you see so these springs that represent microtubules or stress fiber networks, which essentially also transmit forces between nucleus shown by the red and vertices shown by the corners here. And each vertex is associated with a focal adhesion point. And then these vertices can change angle, they can extend, so they can protrude or they can retract. And for that, there's in fact a motor clutch model that is very well established in the literature now, which is incorporated at each vertex. And at the scale of the vertex, so this is what you see here, and then this is the side view of a protrusion. So the motor clutch model includes a variety of components, but the most prominent one is the actin here, which is in the F-actin form, in the polymerized form. But the depolymerization of that would be creating G-actin and then a retrograde flow associated with VR here. But then there would also be a polymerization rate of the actin. And then the difference of that would basically give the spreading velocity of the protrusion locally. So this is not the cell spreading velocity, it's just the spreading velocity of the protrusion. But of course, in the meantime, this structure here interacts with the underlying matrix through focal adhesion points, which are represented by a motor clutch model. So here you see these tiny springs that can form and dissociate with given rates like K on and K off, but they also transmit forces as soon as they are bound, but they can also detach as a function of the forces. And then the underlying substrate most generally would be a viscoelastic substrate, but here we made a simple assumption and then we assumed that the viscoelasticity of the substrate is actually governed only in this context by a Kelvin-Voigt model, which is a very simplistic model. And then I'm actually going to revisit other models in the coming slides. So this is the focal adhesion picture and then the vertex picture. And so this applies to each vertex, to every vertex here. And then there ought to be a force balance at the interface between the protrusion force associated with the actin growth, but also the cytoskeletal force associated with the springs here, and also the actomyosin, the contraction associated with the actomyosin at the interface, and then the elastic membrane forces around the cell, etc. So there needs to be a force balance at each vertex here. But there also needs to be a global force balance at the nucleus here, right at the center. So altogether, like this force balance right at the vertex level, but also at the structure level at the nucleus, will give us a picture of cell, how it migrates and how it changes the shape. So up to this point, I would say this is not entirely novel, but what makes our model stand out from other previous models in the literature is we coupled this model back then with the chemical dynamics of Rho GTPases, the reaction and diffusion of them. And here the label is such that we have membrane-bound inactive Rho GTPases. So this could be like REC and Rho signals. They could be active, shown by yellow here, but they can also be in the cytosolic form, shown by gray. But also within those three states, there's going to be some exchange, and then there are going to be some rate terms associated with that. So there will be some diffusion along the membrane of these molecules, but there will also be a stochasticity associated with them, both with their initial conditions, but also with how they reset at every step that the cell takes. So we are taking that into account, but in particular here, we are relating the polymerization rate of the actin, and this is very well known in the experimental literature, to the RAC signals. And then the number of molecular bonds here at the focal adhesion points, again, to RAC signals. But the myosin motors, when it comes to myosin motors, which essentially amplify retrograde flow, when they are abundant. So they are going to be proportional to the raw signals. Okay, and also RAC and RAW they actually mutually inhibit each other. So they actually, how should I say, space separate, so to speak, such that in a polarized cell you would find a higher concentration of RAC at the front end of the cell and a higher concentration of RAW at the rear end of the cell. And that symmetry breaking also gives rise to the symmetry breaking and the polarization of the cell in the first place. But also here, as I said before, like we had to take some phenomenological feedback from mechanics of the cell to the chemistry. And in that, what we did was, so when a cell protrusion grows, so let's say the angle is not labeled here, but suppose that there's an angle theta here, which is shown by, which is taken to be proportional to RAC. So it will be such that when this angle grows, decreases while the protrusion is growing, at some point there ought to be a feedback from the RAC signal, from the angle of the protrusion back to the RAC signals, so as to inhibit them, suppress their concentration, so that the protrusion is allowed to retract. So we are taking into account all those factors. But of course, like when I say a multi-scale whole cell theory, all parts of this question were previously tackled in the literature one way or another. Like for instance, motor clutch mechanics, there's a very well-established literature on that. So this is only a handful of papers, within maybe hundreds of papers that are on this topic. So there are also whole cell theories, which are not as rich, but there are like different models ranging from phase field modeling to lattice to lattice bolt methods. And so there's also reaction diffusion modeling or signaling dynamics. But here we basically put everything together. So with that, let me give you a pictorial overview of how chemically induced migration occurs, or in other words, how chemistry is coupled to mechanics during migration. So as a reminder, the polymerization of rate of actin is proportional to RAC signals at the front. The number of molecular clutches at the focal adhesion points, so they're assumed to be, again, proportional to the RAC signals on the front. But the myosin motors, either at the front or the back of the cell, so they would be proportional to the rho concentration. And if they're abundant, so there's going to be a strong retrograde flow, which will cause a retraction. And then the idea is this. So let's say this is one edge of the cell which is about to undergo contraction, and here, so the weak polymerization due to strong raw signals will give rise to a strong retrograde flow, a low level of polymerization of actin, but also a weak adhesion with the underlying surface due to the relatively low number of molecular clutches. So all these all together will cause contraction. And here, the phenomenological relation that is widely assumed in the literature is when you have retrograde flow, you can actually assume that to be proportional to the proportional to a term which is like 1 minus substrate force minus actomyosin contraction, which is not shown here, over the number of molecular clutches times the force each clutch can support. So basically, the number of molecular clutches, but then the weak adhesion amplifies this retrograde flow. So there's a coupling, and then the result is a contraction as shown here. So in the case of a protrusion, though, the scenario is exactly opposite. So you can have you should have strong polymerization simply because there will be a lot of RAC signals here, which will amplify polymerization. It will suppress retrograde flow, but it will also create a strong adhesion with the underlying substrate, because in the presence of high RAC, there will be more focal adhesion points forming, and then there will be a more efficient transmission of traction forces from the cell to the substrate. And that will give rise to protrusion. And now when you put these two pictures together, then you can actually qualitatively understand how the cell symmetry can break in the first place in the presence of rack and row polarization, such that if there is rack signal strong here and then row signal strong here, then this part will be more inclined to protrude, and then this part will be more inclined to retract. But of course, every once in a while, that protrusion must stop, and then that retraction should also stop and actually do some momentary here oscillations or undulations in the membrane conformation, as I will show you shortly. So like putting together, so this is actually a movie of a migrating cell, as you can see here. So the red is the nucleus, and then blue is the membrane. So this is in the presence of an initial condition where rack signals are strong here, and then the raw signals are strong on the back. And by using this type of modeling, what we can importantly find is an experimental observation that migration speed varies in a biphasic manner with respect to the substrate elastic stiffness here. So these are the experiments taken from the literature, so these three dots. And there's also other findings in the literature that point to this, this behavior. But we can find this biphasic behavior as a function of cell speed, which is defined as the speed of nucleus over time. And then what you see here is an average over the simulation time and over many cells that we took. So this is the simulation results. And then we can find good agreement with experiments. But of course, the question is, how does this behavior arise in the first place? So here, for this, what we first need to understand is, so we need to define what is soft and what is stiff when it comes to the substrate that the cell is interacting. And so how do we do that? We need to come up with a dimensionless number, or in other words, we need to come up with a characteristic scale of stiffness, substrate stiffness. And that ought to be given within the framework of the cell. So that stiffness we can obtain essentially by setting equal the molecular clutch formation time scale and then the substrate deformation time scale. So this clutch formation time scale is a chemical rate, but this substrate deformation time scale, so it's associated with how the substrate, how the deformation of substrate develops if it's a Kelvin-Voigt material. So it's governed by a time-dependent OD, which we can analyze. So from there, what I can, qualitatively say is, if you have a soft substrate, the traction force takes time to develop on low stiffnesses, and it takes short time to develop on high stiffnesses. But the thing is, so you also want your clutches to be in the formed state, so that you can actually transmit those forces. So then the sweet spot would be that the forces, the traction forces develop in a time that can actually be sustained by the clutches which are on the formed state. So if you take those two time scales, the clutch formation time scale and then the substrate deformation time scale, then you can find the threshold stiffness, which is given by the number of motor clutches, the force each motor clutch can support, and then the clutch formation time scale here, but also the characteristic velocity scale within the cell, which is associated with the retrograde flow. And so this is given to be about 120 nanometers over second or so. So with that, we can actually find a heat map of cell velocity, dimensionless velocity, as a function of substrate viscosity and substrate stiffness. And the threshold stiffness that we determine here is actually so accurate that it gives us a boundary for the termination of the high velocity region of the cell. And then at stiff substrate, like the cell significantly slows down, which actually explains this behavior here. But likewise, just like we can find a threshold stiffness, we can also find the threshold viscosity by setting the clutch formation time scale equal to the viscoelastic force buildup time scale. And that gives us the upper boundary of this high speed region of the cell. So that's one thing. So we can understand the biphasic behavior. But if we consider the symmetry breaking, we can also achieve that just by having a gradient on a substrate, be it in terms of the stiffness or the viscosity of the substrate. Of course, it's very difficult to realize a gradient in viscosity of the substrate, but a stiffness gradient can be fabricated, and there are very mature experiments on those types of setups. So what we can do is we can actually switch off the chemistry, and then we can induce the cell to get polarized by an underlying gradient of the substrate. And here, let me give some definitions. So what I will do is I will introduce a gradient of the stiffness of the substrate here, shown by the grayscale. And then there will be a reference stiffness, which is going to be the initial position of the location of the nucleus. Okay. And then this cell is going to migrate, it'll follow a path, and then we can characterize the motion of the cell from that point on. So here I give you a setup, a movie where the x-axis is in fact the position, and then the y-axis is shown here. So those are the space axis, but also the color code here is such that it follows this substrate stiffness color bar here underneath the movie. And also the membrane force intensity is shown. So this is the traction forces on the membrane. And so there's no chemical signaling here. So the chemical signaling is totally switched off. So that's why you see a circular cell. But importantly, this cell can migrate along the stiffness gradient. So it follows the stiffness gradient from low to high. But here it matters where it starts. Like it starts actually at a low level, which is lower than the threshold stiffness. So the so-called soft stiffness regime. So this is durotaxis, but we can also imagine adurotaxis, and that actually happens when you put the cell initially, put the nucleus initially to a position where the center of the cell is located about the threshold stiffness, so that the cell can't actually decide which way to go, because it could go this way. It's gonna be softer. It could go that way. It could be harder, but it's actually stuck in this regime in such a way that the traction forces on the rear and the front are here balanced, so it can't move. And so this is the adurotaxic behavior. But of course, now you can start the cell from stiffer regimes, from positions where the substrate stiffness is much higher than the threshold stiffness. And then what the cell does is this anti-durotactic behavior. So it starts migrating from high stiffnesses to the low stiffnesses. But then at some point, it gets stuck again. So now we could put together these three pictures. So this is the substrate traction, the traction force of the cell as a function of dimensional substrate stiffness. So this is the intermediate stiffness regime. This is the stiff regime, and this part is the soft regime. And what happens is this: like in the case of durotaxis, basically you have here more traction forces at the front than at the back. So that's how front and back are determined in the first place. And this force imbalance essentially gives rise to a net force in this direction, which pulls the cell towards higher stiffnesses from lower stiffnesses. But then at the stiff regime, you will have the rear of the cell at a relatively lower high traction to acquire a relatively low substrate traction with respect to the front, where the traction forces are higher. So that essentially pulls the cell towards the optimal stiffnesses again. But of course, now if you start the cell from around the threshold stiffness, then the cell will get stuck here simply because the forces at the rear and the front will be balanced, so the cell won't be able to move. So this is in fact a pure mechanics picture of how these effects can occur. But importantly, we can also here consider actomyosin contractility associated with chemical signaling. So we can incorporate into this picture that I just presented the effect of chemical signals, such that when we turn on the chemical signaling and there's some raw signal here, and then some amount of rack signals here for this cell, that's how the protrusions occur in the first place. So now this effect, this mild effect of chemical signals can actually override a previously durotactic behavior or adurotactic behavior, and can pull the cell actually to the left to lower stiffnesses, which would not be preferred by the cell if it were actually only migrating due to the mechanical stimuli. So now we can also tune the signaling, and in this case, for instance, high rho signaling on the back of the cell, and then this turns the behavior completely. So this ought to be the durotactic regime, but if the cell can generate a signaling such that it can override that too, if it likes, or depending on the other external cues, like for instance chemotaxis. So this actually allows us to understand the competition between mechanics and chemistry when it comes to mechanosensitive cell migration. So now this is a picture, but what I actually excluded here in the picture that I showed you so far is an effect which is called adhesion reinforcement, that corresponds to the cells displaying a monotonic rigidity force relationships. And this is simply because a cell attaches to a substrate, and then when the traction forces increase, whereas you would expect that could actually dissociate bonds because of strong forces, because like these springs are now pulled like very strongly, the cells can actually actively reinforce those adhesions by additional proteins like talin, for instance, or other molecules or other proteins as well, and that way can actually generate more substrate traction as a function of substrate stiffness. So they can do it monotonically. But then the challenge here is, so the expectation is that this monotonic behavior in the substrate traction versus stiffness profile should translate into a monotonic profile of speed stiffness, because that's the previous picture that I described. But that's not the case. So what is observed in reality is that the speed stiffness relationship is still non-monotonic, which is a question mark, and it also signifies a non-local effect. Like for instance, so that means that if we take into account adhesion reinforcement, so we cover this behavior, so that's very well understood. But then this question here, or this direction, so that's the main issue that I want to describe next, very briefly for the sake of time. So here the idea is this: so we will take this whole cell, the same cell model that I described. So we will do some modifications to that. For one thing, we will add the adhesion reinforcement effect due to the extra proteins, but we will also change the viscoelasticity of the viscoelastic model of the matrix, and then we will take a standard linear solid model, which is actually known to better explain hydrogels, the mechanics of hydrogels that are used for cell cultures. And of course, again, there's going to be a two-way coupling between mechanics and signaling pathways, as I described before. So there's going to be a local force balance at each focal adhesion site again. But now the difference is that the modern clutch model considers the adhesion reinforcement effect. So we will also take into account the contact mechanics. So I will also present some confinement results between cell and the confinement walls. But importantly now, like to consider the non-local effect, I'm going to hypothesize the following. So what will happen is this. This is the hypothesis. So adhesion reinforcement gives rise to a bigger cell spreading area. So the cell spreads more, which actually squeezes the nucleus. And then that squeezed nucleus is now harder to drag for the cell when it's actually migrating, which is a problem in the case of stiff substrates, because the cell will spread in stiff substrates, but then the price that the cell pays is the difficulty with dragging this nucleus forward. So that can be taken into account by a modified Stokes law, and that's the non-local effect that I'm talking about. And then by using experimental data, we can take into account the dependence of the viscosity on the cell spreading area in the cytosol, but also the shape factor of the nucleus that changes under the increasing spread area of the cell. So now taking into account all these effects, so what we can show is, like, for one thing, this is expected that the cell spreading area increases, and then the substrate force as a function of stiffness of the substrate increases. So those are the results that are expected, that are already put into the model. But due to the effect of modified Stokes law on the nucleus, we can now recover the biphasic behavior of the velocity as a function of stiffness. And that's mainly because the shape factor of the cell, which essentially moves from a circular spherical cell to a more ellipsoidal cell, ellipsoidal nucleus here. So this is the shape of the nucleus. So this is more like a spherical here and more like ellipsoidal here as a function of stiffness, because the area spread, the nucleus is squeezed, and then as a result, so this can hamper the cell speed and then turn the migration speed biphasic as a function of stiffness. But now we can also show that this is really due to this effect, but also due to the shape of the cell, shape of the nucleus, but also the changing viscosity of the nucleus as a function of cell spreading area. So when we switch them off, we can actually recover the monotonic behavior of the cell speed as a function of stiffness as a set of control simulations. So now, as I told you before, like there are cancerous strains that can go between gyrotactic and anti-gyrotactic behavior. Like for instance, these MDA MB231 cells, they can actually switch between adhesion reinforcement and no adhesion reinforcement. So they're able to do that. But in the case of adhesion reinforcement, what we did was we took like 30 cells. So dots here correspond to 0 hours, and then the lines correspond to cell trajectories, and then the triangles correspond to the final position of each cell after they migrate. And then by analyzing these trajectories here, what we could find is that in the case of adhesion reinforcement, regardless of the biphasic speed stiffness relation, just due to the effect of monotonic substrate force versus stiffness relation, this gyrotactic behavior here is actually significantly amplified in agreement with experiments. So all cells like start, so there's a uniform initial condition of cell population on the substrate in simulations, but over time that you can observe that the cell population evolves in such a way that most of the cells migrate to the stiffer region of the substrate. And this is in fact in agreement with experiments, which is shown by the dashed lines here. So like confinement is, as I mentioned before, it's very important because it can change the dynamics drastically. Because for one thing, it suppresses the spreading area, so it actually releases the stress on the nucleus, which actually, like within the context of our picture, would allow the speed to increase, simply because it'll be, again, relatively easier to drag the nucleus ahead with the cell. So, and when we consider confinement, we can actually explain the shift in the experimental observations from a biphasic speed behavior as a function of substrate stiffness to a monotonic speed behavior here. So let me actually show you the effect of confinement. Like here, there are 3 cells, that are racing with each other at different stiffnesses, and then here the substrate stiffness is the channel here. So the channel is the substrate, not the walls, and the stiffness is associated with within the domain inside the channel. And what you see here is, so spreading actually is stronger in a stiffer substrate due to the adhesion reinforcements, but also confinement aligns all the cytoskeletal elements here, such that the traction forces are now directed in a more polarized fashion, and this in fact speeds up the cell. So we can also consider the effect of nucleus, effect of curvature on the migration. So here, what we do is we take into account the curved confinement, and then how a cell curves inside that. So, and then we consider the curvature-dependent membrane stiffness and friction constant, which ought to be there simply because, so the membrane elements are modeled as springs. So on a curved interface, the springs, when they are stretched, they're going to be slightly longer. So you need to take into account that effect, and then if you want to take that into account in a spring model, you actually need to consider the dependence of the stiffness, spring stiffness, but also friction constant to the curvature of the channel. So now, when we take that into account, we can show the dependence of migration speed both to the curvature and how it changes as a function of curvature for changing stiffnesses. Again, like for higher stiffnesses, there's going to be a higher spreading, and for lower stiffnesses, there's going to be a lower spreading. But at the same time, the effect of curvature matters. Like if the radius is smaller, meaning the cell is more curved, the cell is more moving in a more curved channel. So what we observe here is that the velocity is increasing. So sorry, it should be the other way around. I'm sorry. So the velocity is decreasing as a function of channel curvature. And this is simply because high curvature suppresses self-spreading and migration speed. And the reason is there's increasing contact forces in the case of non-linear elastic deformations of a cell within a channel, and that increases the contact forces, which increases the friction forces between the channel and the cell, and then you would observe less spreading due to the increased friction forces, but also a lower migration speed. So with that, we can understand actually in the presence of adhesion reinforcement how non-monotonic migration speed as a function of substrate stiffness is explained. So we can explain the typical MDA MB231 migration dynamics and how it switches between adhesion reinforced and non-adhesion reinforced behaviors. And then we can also show the effect of confinement, like how straight confinement can essentially change the migration speed profile qualitatively as a function of substrate stiffness. But of course, we can also show how curvature matters, and especially how it can amplify the contact forces between the channel walls and the cell, so as to hamper the spreading and the migration speed. So I don't know, do I have time, or should I perhaps stop here? So what do you prefer, Jane?
So you have, I mean, you can stop here. You have 3 more minutes, basically. Maybe we should save some time for questions.
Okay, so let me stop here then.
Okay.
Okay, all right.
All right. Okay, so I see a question in the chat. Ria, would you like to just unmute and ask a question? Oh, no mic. Okay, I can read that out. So
Yeah.
Ria is asking, what are your thoughts on migration of cells which do not have nuclei, like a mature mammalian red blood cell?
Well, I have no idea, honestly. I mean, I think the question would be how you would model the structural integrity in the first place, right? But that's something that I didn't look at, so I don't want to speculate. Also, another thing is, so here we are specifically focusing on mesenchymal migration, but there are also different migration modes. Like there's, amoeboid migration, or there's like lopoboidal migration. So the amoeboid migration, it's more like a, thread milling type of migration, where the cell, sort of starts rolling in a simplistic picture, or lopoboidal migration, where the nucleus basically compartmentalizes the cell and then creates a hydrostatic pressure difference within the cell, and then it can migrate based on that, by utilizing that hydrostatic pressure difference. So it really matters, like what mode is in play when a cell migrates. And a cell, like a mammalian cell, can actually switch between different modes. Like, there's a lot of, strains that can change between mesenchymal and amoeboid mode. And especially like during metastasis, they switch to amoeboid mode, which is actually faster than mesenchymal migration.
Okay, I think Ria said thank you. Sure. Okay, Ria said thank you. Great talk. Any other questions from the audience? You can just go ahead and unmute yourself.
Can I ask one?
Sure.
This is Yi. Okay. The cell is treated as a polygon with n equals to roughly 10, right? Order 10. And then you have to solve the reaction diffusion for rho and RAC and all those internal molecules that dictates the adhesion. How do you solve these reaction diffusion equations with the moving boundary conditions?
So we are solving them on the membrane, which is actually a set of discrete ODs. I mean, it's a PDE, but then you discretize the flux, right? The flux and the diffusion term. But we also take into account the change in the membrane, the change in the length in the membrane sections. So it's basically solved in a Lagrangian frame, the diffusion around the cell membrane.
And you discretize everything in this polygon of n equals to 10-ish. Is that what you did?
Yes, okay, exactly.
Okay.
Yes. So there are like n is equal to 16 vertices, meaning there's going to be like, 16 different values of RAC and rho concentrations each at every given time step.
Got
so
it.
are you? So I have a follow-up question for that. So you're basically saying, okay, the diffusion or any kind of a transport flux of the rho and RAC from one edge to another edge, and that's going to be scaled with the length of the edges.
Yes.
Right.
I mean, so yeah, at every step, like the scale, like the flux term has to be scaled by the length between two mesh points, which are the edges here. So we take that into account here. So it's actually, in a material frame.
So it's never solved within the cell. It's only at the vertices.
No. Yeah,
edges.
I mean, so yes, so that's the shortcoming of the model. So we assume that it instantly equilibrates in the cell, because there's also evidence that it's not purely diffusive inside the cell. So we assume that the concentrations of RAC and Rho inside the cytosol are instantaneously equilibrated, and there's only a rate equation that is coupling the concentrations from inside the cell to the membrane. So there are only rate terms, no diffusion terms inside the cytosol.
Thank you.
I also have a follow-up question for that. So, because you know, so the segments are kind of lining out the profile, kind of on the plane. But I guess you know the actual membrane, you also have like the upper
Right.
and the
Right,
lower,
yeah,
you know, the top and bottom. Like,
Right. Right.
how does the scaling work to like sort of lump all those effects together?
Right. So this is a 2D model. But when it comes to squeezing of the nucleus, so there we consider shape factor. I mean, so basically there's a height of the cell associated with its initial configuration, and then from there you assume mass conservation, volume conservation inside the cell, and then from there you can essentially solve for the changing aspect ratio in the z direction when the cell spreads. And then from there, you estimate, you know, how much the nucleus should be squeezed, at least its order of magnitude. And then how that should change the shape factor, in turn modifying the Stokes drag.
I see.
But yeah, there's never really a third dimension in this model. So it's
I see.